Imagine for a second a future where we don't just observe biology, but we can actually ask it questions. A future where we can map out the incredibly complex cause and effect circuits inside our own cells, not just what they are, but what they could be. Well, that future, it's not so far away. In fact, it's starting to happen right now, thanks to a radical new partnership between AI and biology.
This quote, it really gets to the heart of it. This isn't about AI replacing scientists. Not at all. It's about empowering them. Think of it like giving every researcher a collaborator that has the creativity of a human, but also the sheer analytical power to just crunch through mountains of data and explore possibilities we couldn't even dream of. It's all about augmenting our own ingenuity, not replacing it.
I mean, really think about that. What if you could predict what would happen in almost any genetic experiment before you even set foot in a lab? What if you wanted to know what happens when you turn off gene A, but also crank up gene B at the exact same time? This is the central promise here, the ability to run a nearly infinite number of biological experiments, all virtually.
Now, to really get why this is such a massive breakthrough, we have to first appreciate the monumental challenge scientists have been up against for decades, and that is the sheer, overwhelming complexity of our own biology.
You know, a single one of our cells is like a tiny, bustling city, with thousands of genes all interacting in these complicated, unpredictable ways. These little circuits, they determine everything about us. But trying to map them out, to really understand the cause and effect, has been unbelievably difficult.
And this number, well, it shows you the mind-boggling scale of the problem. If you just look at combining 2 genes out of our 20,000 or so, you're already at nearly 200 million possible combinations. If you try for 3 genes, you're talking over 1 trillion. Trying to test every single one of those in a lab is, well, it's just impossible.
This is what scientists call the combinatorial explosion, and for a long time it's been the Great Wall of biology. For decades, the idea of a complete causal map, a true wiring diagram of the cell, seemed like a distant dream, something we might never achieve. But what if you didn't have to test everything? What if you could run way smarter, way richer experiments at a scale we've never seen before, and then let an AI fill in the blanks? This is where the breakthrough really begins, with a revolutionary new toolbox. So a key piece of tech here is called Perturb-seq, and it's brilliant. Basically, it's a way to go into a cell and poke a specific gene using tools like CRISPR, and then immediately read out all the ripple effects across the entire cell by looking at its RNA. The best part? You can do this for thousands of different genes all at once in a single experiment. It's a total game changer.
And this gives you a sense of just how sophisticated this toolbox has become. We can now apply these perturbations, these little genetic pokes and edits, across a whole range of biological systems. We're not just talking about simple cells in a dish anymore. We're talking about complex organoids that actually mimic our organs, and even seeing these effects in whole animal models.
The old way of doing biology was kind of like looking at a single light on a massive circuit board. You could see if it was on or off, and that was about it. These new methods, they give us a high-definition 3D picture of the entire board. We can see the full profile, we can group genes into functional teams, and we can finally start to understand how the cell is actually wired, not just what happened when we flicked a switch.
Okay, but generating these massive piles of data is really only half the battle. The other, equally important half is figuring out what it all means. And that's where AI comes into the picture.
This is just the perfect partnership. You have these new experimental tools generating enormous, rich data sets of cause and effect. And then you have AI models, which are perfectly suited to learn the underlying rules of biology from all that data, to find the hidden patterns in all that complexity.
And this is where it gets really, really cool. Models with names like Gears and SkepGPT are being trained on all this data. You can think of them like specialized students of biology. Some use a technology called graph neural networks, which makes them experts at mapping relationships. Others use tech that's pretty similar to what powers ChatGPT. They learn the language of the cell so well that they can actually start to predict the outcomes of genetic changes they have never seen before.
So, this creates this incredibly powerful feedback loop called active learning. You run experiments, that feeds data to the AI. The AI then makes new predictions. Then scientists can run just a few very targeted experiments to check those predictions. That new data makes the AI even smarter, and the whole cycle repeats, getting faster and more efficient every single time.
So where is all of this going? What's the endgame here? Well, it's leading to something truly transformative, a completely new kind of map for human biology.
I love this comparison because it just clicks. For years, we've been building something called the Human Cell Atlas, which is this amazing project to map every single cell type in our body. Think of that as a beautiful but static street map. This new vision, a perturbation atlas, is more like Google Maps for our cells. It's a dynamic, predictive tool that can show us how to get from a disease state to a healthy one, what happens if a genetic road is closed, and maybe even how to engineer a totally new biological route.
And the real-world impact of this could be profound. I mean, with a causal map like this, we could finally figure out what genes linked to diseases actually do. We could design cells for new therapies, find better drug targets, and predict how combinations of drugs might work or not work long before ever starting a clinical trial.
So the ultimate goal, the grand vision, is to build a single, unified foundation model of our cells, one that's generative, meaning it can predict and create new states, and causal, meaning it actually understands the fundamental wiring diagram. This would truly be a new atlas for a whole new era of biology.
You know, we're moving from just reading the book of life to actually being able to write new sentences. We're learning how to have a real conversation with our own biology. So the question for all of us is, when we can finally ask ourselves anything, what are we going to ask them first?